for meeting you. We are now talking a little bit on sort of the historic in your development in the biopolymer research area, as well as what is going on in the future. Together with me, we have Professor Kurt Draget and Catherine Taylor-Norgård. Kurt, when it comes to biopolymers and the sort of development of your research and this invention of the Makova or G-block, how was that sort of developed over time?
Well, to take it from the very start, it was actually a handful of guys sitting down in London just after the World War, trying to figure out what should we live of after the war. And someone came up with the idea that we had so much brown seaweeds that it should be possible to do something with that. So based on that, it was actually established an institute here in Trondheim called the Norwegian Institute of Seaweed Research, which actually started out as a pure basic research organization directly under the Norwegian Research Council and not as a department at the university. So they were actually funded for 20, 30 years directly without having any requirements for commercialization. So that's actually what we are working, that's our work is based on now. Is there 30 years of basic research.
So we're talking about 60 to 30, how long? 60 years?
60 years, yep. Well,
amazing.
Well above, yep. So that's when they discovered, or in collaboration with other partners as well abroad, the composition of the alginate molecule from the brown seaweeds. And that's why we now today know exactly how we shall get hold of these G-blocks, which are components of the alginate molecule.
So it's synthetic, or how do you develop your G-block?
No, this is purely of natural origin, because these are saccharides, oligosaccharides.
Right.
And different from proteins, they cannot be easily synthesized. It's actually not quite even, it's extremely challenging to synthesize a oligosaccharide, not to mention a polysaccharide, because you have so much more options for creating covalent bonds compared to, let's say, a synthetic protein, which is much more simple.
Okay, and when you look into the development of the G-block as such, when did you sort of define it and find the effects of G-block versus
this was actually, now we're entering to what's it started out as a hypothesis because Catherine had, during an earlier industrial project some 10-15 years ago, discovered that there was a synergistic interaction between mucins in mucus and alginates.
Okay.
And in the indication called cystic fibrosis, you always get an infection in your lung that produces alginate. So we had the original hypothesis that if we isolated small fragments of the alginate molecules, that could sort of prevent the interaction between the alginate from the bacteria and the lungs and the mucins, and thereby reduce the viscosity of the mucus. And it worked. But then serendipity enters into the picture, because we also found that the G-blocks were able to counteract or act in between mucin-mucin interactions. So we could actually, even non-pathological mucus could be modified with these G-blocks.
And that's when we came into the drug delivery field, because here you're talking about a normal mucus that we want to modify to change the barrier properties.
And we're talking, when was that discovered in?
That was first patented in 2005.
2005.
Yeah.
And that was from this institute?
Yes. Yeah. Absolutely.
when you are, I'm just sort of thinking as a researcher, when you look into that sort of discovery and you're going to consider commercialization and you take the tech trans organization and you start patenting and all that, I know that there's a lot of confidentiality. You cannot sort of start up your publications in time, and as a researcher you're often sort of measured on publications, citations and so on. Is it frustrating to live with that, or how do you deal with that sort of inventions?
Well, I think we can live pretty well with it, but of course it's on collision course with the classical university,
right?
Which is supposed to be an open institution, publications as you said, and there's how shall I put it? It's not very well understood by everyone working in the university system how these commercialization processes actually proceed. So it's as I said, it's slightly contradictory to the classical university and openness, but I mean we can live with it, and it's also some sort of the philosopher Plato with everything is done for a reason, right?
Right.
So we feel the same, that if we see something that can be applied, let's say in medicine, no one else can take the responsibility for that. We have to do that.
Right.
It's something of an ethical responsibility, because if you're in your lab and you make a discovery that you believe could be used to better human health, you have, as an academic researcher, a choice. You can publish it, in which case you guarantee that it's not going to be able to be commercialised, it's not going to be able to be used to better human health. Or you can say, okay, I'm going to do what I believe is the right thing, and say, okay, we have to look after this, we have to make sure that we act in a way to enable it to be used.
Right.
And that means, in the current situation, it means patenting, which obviously we contact our technology transfer office, we talk to them about the process, and once we've patented, that's not enough. That's not the final product. You actually have to continue to work on development, on getting this through the tech transfer process. The idea of transferring it out of the academic world and into, well, we can say, the real world. And that's something that has an ethical relevance. If I was to simply publish this, I have guaranteed it cannot be used. There is no pharma company who's going to take published data, which anyone has access to, and spend all that money on development.
That's right. Yeah.
So we have an ethical responsibility, and whether or not...
And you need that collaboration with pharma.
Absolutely.
To bring it up.
How much does it cost to develop a medicine? This is not something we can do from an academic lab. This has to be a collaboration.
When I talked to you before, we talked about the tech trans department and the way they help you in money-wise as well, the finances. So even though it's restrict you to a certain extent when it comes to publications right ahead, you still have support in finances as well, bringing up more research from a commercial point of view.
Yes, absolutely.
Yeah.
Because, well, at least in Norway, and I would be surprised if it wasn't valid for the rest of Europe, the tech transfer organization at universities has some fundings, or easy access to fundings, that normal, should I say, well, researchers normally don't have access to. Which are outside the typical National Research Council, which brings us closer to getting funding reasonably safe and reasonably fast,
Right.
which is very important in this context, because once you file the patent, the clock ticks.
Yeah.
And you have to remember, it's not actually just about financing. also through the Tech Transfer Office, we come in contact with these industrial partners, with people who actually have a very good understanding of the problems that they're working with. And that informs our research as well.
Okay.
Because if we're going to work on real world problems, if we're going to try and deliver solutions, then it really helps to be in detailed contact with people who have the problems. Rather than just sitting there in our lab in an isolated sense, this is a contact net out into the commercial world with people who can tell us more about the problems they're facing and enable us to deliver the kind of solutions that will address these problems.
So actually the tech trans department here at the university is spearheading the pharma companies and trying to develop license agreements and MTAs and so on.
Yes, that's
Okay.
correct. Mm.
But you're still in the process or as a researcher,
Oh,
or can you
absolutely.
sort of...
Absolutely, because I must say, for those of us who likes to put things behind us, you should never do patenting, because it comes again and again and again after the different examiners at different national levels. So it doesn't stop.
It's an ongoing process.
It's a very ongoing process, yes, absolutely. So our main responsibility in that context is that for usually, when you file a patent, you go out quite broadly. But then you have to narrow claims, right?
Yeah.
Depending on what's been found during the examination process. So our main responsibility then becomes to make sure that by limiting the claims, we don't end up with something that's a piece of paper. It's still got some context, it's still got something worth protecting.
And there's also the element of communication as well, because if you just have your tech trans people going out and talking to companies, and then they get a question, well, can we use it like this?
Right.
What will happen with this? Then suddenly you're going through multiple links
Yeah.
to get that information. If you've got the researchers on board, then you can go straight to the source. And I can be sitting on the telephone with someone from a pharma company and they say, could we use it like this? What would happen with this? And I'm there as part of the process, actually able to answer those questions right at the beginning. But of course that means that we do have to spend time as academics learning about this process. Because we can almost say there's two different languages, the language of academia and the commercial language. So you have to become bilingual in a sense, so you can actually address the concerns of industry and deal with the questions that are coming in and really get that communication going.
When I did research, there was sort of a saying that when you're collaborating with the big pharma companies, they were sort of taking a lot in, but they're not sort of giving in information the other way. What is your experience in that process, and what is your do you work with big pharma companies today?
Yes, we're doing that on several levels, both as well in MTAs and licensing from tech trans organization, but we're also into an IMI, which is Innovative Medicines Initiative, which is a EU Framework 7 program,
Right.
in which you have a collaboration between universities, small biotech companies and large pharma actors. So we have a good contact net with big pharma on several levels.
How does it work when you're considering your confidentiality and the IP package and all that together in that forum? Is that something you need to be concerned of, or do you have sort of a game plan?
No, absolutely. I mean, what we bring into the project,
Right.
into this EU IMI project, is our background. But obviously we have to make sure that we don't mix things up, that our commercial process going on here should not or shall not enter into the compact project, because you have to make it really separate.
Okay. As the professor of Biopolymer Institute, and how do you look into the, both of you, how do you look into the future of your research, and will you do similar, will you focus more on collaboration with big pharma, or which way are you going in the future?
Well, we have to, won't we? I mean, if you're going to get this to the human clinic, we can't do that on our own.
No.
Really, because of the resources needed.
Yeah.
Thinking about man hours and money. So it's a prerequisite actually. If we're serious about getting this out to the benefit of human beings, we have to collaborate.
Right. And your pipeline, how sort of are you developing new things in the future, or is you sort of building up on the G-block technology, or what is your thoughts about that?
Well, we hope, but obviously, I mean, you can't plan inventions, can you? Because then it isn't an invention.
That's right.
If
So
it was obvious, then
that's not possible. So, but for the time being, we are more than preoccupied, I would say, with the G-block technology and sort of unraveling the potential of it, which seems to be rather unlimited for the time being. We can't see the end of it. So there's a huge number of possible pharmaceutical applications they can be used for.
Well, I will say thank you very much for your time. It's been extremely interesting to hear about G-block technology and the platform. Thanks a lot.
Thank you.